Alright, this video looks at influenza. Alright, this influenza is the influenza that you should be getting a vaccine for each year. And what I would like you to take away from these few PowerPoint slides is the disease process, what goes on when someone gets the virus, contracts the virus. Be able to describe genetic drift and shift, as these are very important to the reason why we need to get yearly vaccines, and why every once in a while, say every 10 years or so, we have a major issue worldwide with influenza. And then also recognizing the surface proteins on influenza that are helpful in their own quest to infect cells, as well as the role of those proteins in creating a vaccine. So here is a picture, a very famous picture of influenza and how many people it can affect very quickly. And if I am correct, this is a picture from when the Spanish flu of 1918 hit the United States. And this is a YouTube link for a very interesting video, and I'll put this on the website directly so that you can go to this and watch this 8 to 10 minute video on the history of the Spanish influenza. But influenza itself is a viral infection, and the signs and symptoms of this are upper respiratory tract related. However, if it is a bad case of influenza, it could actually progress down to the lower respiratory tract and maybe even cause pneumonia. But if you have just plain old influenza, then your signs are going to be headache, chills, dry cough, body aches, fever, stuffy nose, sore throat, all of those good things that sometimes is caused by the virus itself, but other times caused by the body's immune response to that virus being inside of us. And then there's also extreme fatigue that can occur with this. So with differences between influenza and just a bad head cold is usually the fever and the extreme fatigue. Generally with the cold virus, unless you're a young child or an infant, you don't typically see a fever. And the fatigue is usually very different, right? Sometimes with a head cold, you can go out about your day for a couple of hours or even all day, and then by the time you get home at night, then you feel like you've been hit by a bus. Just that extreme fatigue where you can go to bed and stay in bed, going to bed around 7 o'clock at night, which might be pretty unusual for most of us. But with influenza, you may not get out of bed in the morning to actually do anything that day. That's how fatigued you can be. And then also with influenza, especially if you have underlying health issues or you have a suppressed immune system, you are at risk more so than a healthy, immune competent individual of contracting secondary infections. And the secondary infections are then generally what's going to be problematic for the lower respiratory tract. But those secondary infections usually add different signs to the course of the disease. So like with a dry cough, it's dry cough, it's just influenza, but maybe with a secondary infection, then that crosses over to a productive cough. Alright, now the causative agent for influenza is going to be influenza virus A, B, and C. But for test purposes, you're going to want to know that it comes from the Orthomyxoviridae family. And that is the group within, or that houses influenza viruses. And this talks about ion channels and single-stranded RNA genome. But really, besides the causative agent of Orthomyxoviridae, I want you to focus on the hemagglutinin and the neuraminidase. These are viral spikes, glycoprotein spikes, that sit on the surface of the virion. All right, so these little pink flat diamonds are the neuraminidase, and then these funky mustard-colored receptors are the hemagglutinin, so H and N. These are important because they can be used for the production of vaccines because they are on the surface of the virion. Once that virion gets into our body, that's what the host immune system is going to see. So those two proteins can be used in a vaccine to activate our immune system, so as soon as that virus gets in, it is recognized by the immune response and it clears it before the virus is able to get into our cells and cause major damage or disease. Alright, the other thing is that the H and the N actually have their own nucleic acid sequence, alright, their DNA sequence. Even though it's RNA, it has to get, well, it's going to be used to make protein, but those can be very different from one virion to the next. So you have different serotypes of the virus. And so if you hear about influenza in the news, you might hear, well, H1N1 is a problem, and H5N10 is a problem. And the H and the N, when they're using that nomenclature, they're using the genes found in the virus. And so there are many hemagglutinin versions, and there are many neuraminidase versions of the gene. And differences in the genetic material translates to differences in the protein. So that's why it's important to get a vaccine every year, because these surface proteins at the nucleic acid level are going to mutate randomly and cause slightly different proteins. And so you want your immune system to be as primed and ready as possible if it encounters the virus. And so when you hear H1N1 or whatever the nomenclature might be, that's what's happening. That's how they're describing that actual virion that's causing the problem in the population, by looking at the sequence of hemagglutinin glycoprotein and the sequence of the neuraminidase glycoprotein sequence. So with influenza, and actually with a lot of different viruses, but influenza has made this concept famous, so to speak, is antigenic drift and antigenic shift. So you have influenza that actually mutates at a pretty high frequency, and it's really the hemagglutinin and the neuraminidase genes are what's paid attention to. And as those genes mutate, the proteins look different, and so that's going to reduce the host immune response to the virus. And that causes problems, right? Because our immune system can't fight it because it looks different. And the antigenic shift is just by minor mutations, random mutations, and that produces most seasonal influenza strains. So what is going to cause a problem this year was probably viruses that came around last year, but then mutated over time. And so now there's a slightly different version out there that can cause a little problem. With the antigenic shift, this is when there's actual RNA or nucleic acid exchange between two different viruses. And what has to happen here is you have co-infection of two different cells or two different viruses in one host cell. And the antigenic shift is what generally causes most pandemic strains. So the strains of influenza that are problematic across continents. All right, jumping from Asia, which is generally where these strains originate, maybe moving through Europe and into America, or Asia into America. And if you think about the drift versus the shift, All right, drift, if you think about being in a pond or a river, and you just kind of drift along with the natural flow of the water. You don't necessarily go a big difference or move a big difference in a short amount of time. You're just kind of drifting. Whereas if you shift, you're moving from one pond to a next. And so therefore the change is a major change. And so here's one picture. This is the picture out of your textbook that diagrams, just checking to make sure the other picture was there, that diagrams this idea between shift and drift. And this is looking at shifting, where you have the duck influenza virus here, so it's in blue, and you have the peach colored influenza virus here. And this is duck, this is human, and in close scenarios, or in scenarios where there's close proximity between a duck, a human, and a pig or swine, all right, these two individual viruses can infect the pig, and if both viruses are able to get into the same cell of the pig, then you have this brand new, really brand new virus here. All right, human influenza virus with duck hemagglutinin spikes. And so therefore, that causes problems for this person because half of this virus, half of what the immune system is seeing is brand new. And so the immune system just doesn't know how to respond, and it doesn't, and this person gets very sick. All right, I like this version from a different textbook a little bit better. And what it shows is genetic shifting here, where you have this shade of purple or blue virus going into the cell, and there being some minor mutations while it's in this cell, in this host, and then the new virus comes out, and it's just a slightly different shade of purple. So not too big of a difference, but enough to cause, minor issues. And you see the number of cases are relatively even. All right, every other year having outbreaks. Over here, though, what you have are virus 1 and virus 2, red and yellow, infecting the same cell at the same time, and you have the mixing of the genetic material. And so then what do you get? Well, what do you get when you mix red and yellow? So yeah, red and yellow paint, all right, as you might have done in grade school, kindergarten, you now have orange. And so this is an orange virion, and it looks very different than these two. And so this is a major issue in terms of causing virus to be basically released out into the world where no one has seen this virus before. And what then occurs is you have the pandemic strains, where this is what you see under normal scenarios, but when this occurs with this genetic shift, you have major outbreaks. And this is what occurs about every 10 years or so. And this portion would be the pandemic, where it's crossing continents and causing a lot of problems. All right, and then finally you just have your disease table here. Looking at the causative agent, again, remember that influenza for your testing purposes is caused by orthomyxoviridae. And virulence factors are just those glycoprotein spikes. You contract it by droplet contact, direct contact, indirect contact, basically by the mucus, all right, whether that is blowing your nose and you drop in your Kleenex and someone comes along and picks up your Kleenex and doesn't wash his or her hands and then touches his or her eye or nose. All right, you've got self-inoculation there. Or you sneeze and you aerosolize the virus and someone in close proximity sniffs your sneeze material and they have then contacted the infection. And that is it for influenza.